Hi Mansumala. Today I am going to discuss about the gene expression. Gene expression is the process by which information from a gene is used in the synthesis of functional gene product. Gene control of protein. The type of RNA that contains the information for making a protein is called messenger RNA because it carries the information or message from the DNA out of the nucleus into the cytoplasma. Translation, the second step in getting from a gene to a protein, takes place in the cytoplasma. How do gene direct the production of protein? Through the process of transcription and translation, information from the gene is used to make protein. Transcription transcription is the process of making an RNA copy of a gene sequence. This copy called a messenger RNA molecules. Molecules leaves the cell nucleus and enter the cytoplasma, where it directs the synthesis of the protein which it includes. Translation is the process of translating the sequence of a messenger RNA molecules to a sequence of an amino acid during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes in the cell cytoplasma. The ribosomes reads the sequence of the mRNA in group of 3 bases to assemble the protein. A requirement for protein synthesis: 1 DNA, 2 messenger or tRNA, transfer RNA, ribosomes, amino acids, enzyme. The main protein synthesis steps are initiation, elongation, termination. Initiation. The first of protein synthesis is initiation, that cover the assembly of translation synthesis components and proceed the formation of peptides bonds. The components involved in the first step of protein synthesis are the mRNA to be translated, the 2 ribosomal subunits, the amino acyl tRNA, which is specified by the first codon in the mRNA, guanosine triphosphate, which provides energy for the process. Eukaryotes require also adenosine triphosphate. Initiation factor, which enable the assembly of this initiation complex. Prokaryotes have 3 initiation factors are known: IF1, IF2 and IF3, while eukaryotic there have over 10 factors designated with EFI prefix. Translation elongation. Translation elongation is the second in the protein synthesis step. During the elongation step, the polypeptide chain added amino acids to the carboxyl and the chain protein grows as the ribosomes moves from the 5' to 3' of the mRNA. In prokaryotics, the delivery of the amino acyl tRNA to ribosomal A site is facilitated by elongation factors EFTU-GTP and EFTS and required GTP hydrolysis. In eukaryotes, the analogous elongation factors are EF1 alpha GTP and EF1 beta gamma. Both EFTS in prokaryotes and EF1 beta gamma in function as nucleotide exchange factor. Translation elongation. The peptidyl transferase is an important enzyme which catalyzes the formation of the peptide bonds. The enzyme activity is found to be intrinsic to the 23S RNA found in the larger ribosomal subunit. Because this RNA catalyzes the polypeptide bond formation reaction, it is named as ribosome. Termination. Termination happens when the site of the ribosomes reaches one of the three termination codon.